So if you recall, in the last section, we talked about the first step of getting from genetic information to a protein, and that is called transcription when we go from DNA to RNA. So today, we're going to talk about what's known as translation, and this is where we go from the RNA to the protein. And if you remember, going from the nucleic acid to a polypeptide, these are two different chemicals or macromolecules, and so that's what requires that language to be translated. So the cell basically is going to read the genetic code and build a protein from that, and that message is found in the mRNA. So it does that through the help of something known as transfer RNA or tRNA, and tRNA acts as the translator. And the main job of tRNA is to decode the mRNA codon and deliver the amino acid that corresponds to the mRNA codon. So it kind of is responsible for bringing the correct amino acid to the codon after reading the code. Then it will bring that to the ribosome, and the ribosomes will help catalyze the formation of peptide bonds in order to form amino acids, and then finally the protein. So this is kind of like the whole picture of what's going on in translation, but we're going to start just with tRNA. So each of the tRNA is able to translate a particular mRNA codon into a given amino acid, and its structure really influences its function. Once again, common theme in AP Bio. So on one end of the tRNA, there's an amino acid, and at the other end, there's a nucleotide triplet that is complementary base pair-wise to the codon on mRNA. So they kind of look like a clover if you look at them in a 2D formation. And then this is a little bit what a 3D tRNA would look like. But they bind to particular amino acids by a covalent attachment at the 3' end of the tRNA molecule. So this is where our amino acid is going to attach. They also bind to the mRNA, and this happens around the middle of the tRNA at what's known as the anticodon. And this is a series of 3 bases that is complementary to the mRNA codon. And then finally, the tRNA can interact with ribosomes. And so its structure allows it to interact with ribosomes. So this is what it kind of looks like in 2D form. In 3D form, this is where the amino acid would attach. And then over here is where the anticodon is. And then in your textbook, you're going to see the tRNA looks a little more like this. That's how they're going to draw it. So really when we're working with our tRNA molecules, they also work very closely with ribosomes. And this is the ribosomes are kind of the molecular workbench of translation, so that's where all of the translation happens. And in both prokaryotic and eukaryotic cells, ribosomes are made up of 2 subunits. So they have a large subunit and a small subunit. And the large subunit in eukaryotes consists of 3 rRNA molecules and 49 protein molecules. So there's a lot of stuff going on in the ribosomes. Within the ribosome, there are 3 different sites where the tRNA can bind, known as the A, P, and E site. This A got a little misplaced, it looks like, when this PowerPoint transferred over, but this would be the A site. And so when we're taking a look at our ribosome, you can see we have a small subunit and the large subunit, and then we have our A, P, and E site. This is your mRNA that's going to be going through translation to make the protein, and then this is the growing protein leaving the ribosome. So the A site stands for the amino site, and this is where once the tRNA picks up an amino acid, it's going to bring it to the A site and bind. Here is where it's going to bind to the mRNA codon. So it will line up the correct amino acid that needs to be added to the chain, and then in the next site, the P site, This is known as the polypeptide site, and this is where the tRNA is going to add amino acids to the polypeptide chain. So this is where we kind of get lined up, and then the P site is where the amino acid is actually added. And then finally, once that's been done, the tRNA is now empty because it's given up its amino acid to the polypeptide chain, which is going to start to leave eventually. And that now empty tRNA is going to move over to the E site, which stands for the exit site. And this is where it prepares to be released from the ribosome. And then once it's released, it's going to go back into the cytoplasm. This is the tRNA, gets released, goes back into the cytoplasm, and picks up another amino acid. So this kind of shows you what is actually going on. We've got our amino acid coming to the A site, getting lined up, or a tRNA coming to the A site, and it has the next amino acid. It's ready to be added. That pairs with the mRNA. Then in the P site, that's where some reactions are going to happen that create a bond and add this amino acid to this chain. And then once that amino acid has been added, he's now empty and can go back and retrieve another amino acid. And remember, these tRNAs are going to retrieve the amino acid that has the corresponding anticodon to the codons that code for different amino acids. So once again, in words, the P site holds the tRNA that carries the growing protein chain. That's the second one. The A site holds the tRNA that carries the next amino acid that's going to be added, the amino site. And then the E site is the exit site. So once the tRNA has left its amino acid behind, it gets prepared to exit the ribosome. So I'm actually going to stop here, and then I'll explain the rest of translation in the next video.